Okay, then I'll start. So hi, as Philipp just mentioned, my name is Laura Villegas, and today I will be talking to you about this project that I've been conducting at the University of Cologne on the topic estimation of mutation rates in asexual and sexual panagrelymid nematodes. So first, I would like to tell you the structure of the talk. I will start talking to you about nematodes, which is the subject of the study and what was our research question. Then we will talk a bit about the experimental design, the data analysis, which is where we actually estimated the mutation rates. And finally, we will talk about what these mutation rates mean in terms of sexual and asexual reproduction. So first, let's talk about nematodes. Nematodes have been reported in every continent, and they can either be free-living or parasitic. And here we will be talking about free-living nematodes, and this means that they can be living, for instance, in water bodies or in soil, and they have been reported, as I mentioned, in every continent. This also includes really extreme environments, such as Antarctica and deserts. They can also withstand really extreme conditions, such as high salinity and low moisture content. They play really important roles in ecosystems as they are decomposers, predators, they are food for higher trophic levels, and they also act as bioindicators for climate change and ecological disturbance. They are really interesting as well because they can reproduce in different ways. So they can either reproduce sexually or asexually. This means they can reproduce through amphimixes, hermaphroditism, or parthenogenesis. Which raises the question, why are there so many different types of reproduction, and is there not just one that is optimized for all organisms? Well, sexual reproduction is almost ubiquitous on animals. This means that it's the one that is mostly present on all of them. And it comes with several benefits, such as recombination, it's aiding the purifying selection against deleterious mutations, and here genetic diversity can be maintained. However, it also has some trade-offs, such as the twofold cost of sex, as well as the possibility of acquiring, for instance, sexually transmitted diseases. On asexual organisms, there's no need to find a mating partner, which might be the reason why asexual nematodes can thrive on extreme environments. However, there are also some proposed trade-offs, such as the reduced heterozygosity and the accumulation of deleterious mutations. Several taxa have already challenged these assumptions, and here we would like to talk about mutations. So can the accumulation of deleterious mutations be prevented or dampened at all? Mutations are changes that occur across a nucleotide sequence, and most of the mutations that occur are deleterious, which means they have a negative effect on fitness. And in the absence of recombination, which is the case of asexual organisms, then they are expected to accumulate, which will cause then Müller's richet, which can lead to unviable genomes. How often mutations are occurring, and this not only refers to deleterious mutations, but also to neutral mutations and beneficial ones, is known as the mutation rate, which we refer to as the number of mutations per site per generation on a genome. And there are several theories that take place in terms of asexual reproduction and mutation rates. One of them is that if they occur less often, this means a lower mutation rate, then more generations will be needed for a considerable decline in fitness to occur. And if they occur more often, this means a higher mutation rate, then there's a higher chance of beneficial mutations to occur. And this brings us to a research question, which was, is long-term genomic stability of asexual species due to lower mutation rates? And to test this hypothesis, two different strains of nematodes were used. One is a Panagrelymys nematode, which is asexual and was isolated in the United States. This is a parthenogenetic species, which means eggs are not being fertilized. And the other one, which is Propanagrelymys, is a sexual one that was isolated in China, and it's an androdiecious species, which means it has hermaphrodites, and this means that they produce both egg and sperm. So the experiment was set up as I will just explain to you, but I was not conducting this experiment. I just used the data coming from it, and this was performed by Philipp on his doctoral thesis. So the mutation accumulation line experiment starts with one nematode that it's allowed to reproduce and it's sequenced as a reference, so a starting point. And then single nematodes are then placed on fresh agar plates, and each of these plates with one nematode becomes the start of one line. And in this case, there were 100 lines for the asexual strain and 71 for the sexual one. Several generations are allowed to pass, and on each case, one nematode is transferred from a previous plate to a new one. And the end point of the experiment then is resequenced again to compare to the reference point, so we can see how many changes occurred through time. In this case, Illumina sequence was used, and the final lines had in between 30 and 52 generations. And also a system of backups was allowed because also strongly deleterious mutations can occur, which would mean the end of a line or even crawling of the nematodes out of the plates. Now here comes what I actually did in the project, which was starting with mutation calling. So we used 6 lines for each of the nematode strains. And the process began with pre-processing of the data, which is basically mapping, trimming, removing duplicates, and getting rid of low quality reads. Then we estimated several parameters for instance, the GC content on the reference genome and Watterson theta for both of the strains. And these parameters were estimated because they were required for the tool accumulate, which is what we used to then obtain all the candidate mutations that had occurred. And here what happens is that it looks at the transition of alleles from the central state, so this starting point where we sequence the reference, to the descendant mutation accumulation lines. And this gives us a result, a high number of mutation candidates, which we then filter through several steps. For instance, we look at coverage ranges, we check that mutations are only present in one of the lines, and that they are not present there, for instance, because of errors in mapping. So then what we did is, with the final number of candidates after filtering, we estimated mutation rates just following this equation, the number of called mutations divided the number of generations times the callable sites. And this was performed for each of the lines of each strain. And then we just estimated an average for each strain of nematodes. And what we obtained was a lower mutation rate for the asexual strain. And both of the mutation rates that we found here were lower than that estimated for C. elegans, which is the most prominent nematode in research. And I want to highlight something here, which is that due to the low number of de novo mutations that we found, there was no statistical significance between the difference of both mutation rates, but in general, there is a tendency between the ranges for the asexual one to be lower. So what does it mean then if we have a lower mutation rate in the asexual strains? As I mentioned at the beginning of the talk, if they are occurring less often, so a lower mutation rate, this would mean that more generations are needed for a considerable decline in fitness to occur. In general, for all organisms, mutation rates are expected to be low to prevent deleterious effect on fitness. And now specifically talking about asexual organisms, it has been reported as recently as last year throughout simulations that low mutation rates can maintain existing favorable effects because fewer mutations are occurring that can hinder the beneficial ones. Then when mutation rates are very low, advantageous traits can evolve slowly. Whereas if we have high mutation rates, the opposite occurs. Negative effects of deleterious mutations are likely to overcome the advantage of the beneficial ones, as I just mentioned, and then advantageous traits are unlikely to evolve. Also, a process known as clonal interference can occur, which is that if many beneficial mutations are occurring in a population, then those individuals that have them would be competing against each other. And this means that these beneficial traits will not be fixed as fast as they could have if it was a sexual reproduction population, as we can see on this image on the left. So in general, we could say that lower mutation rates are advantageous not only in terms of maintaining existing favorable effects, but also in acquiring new ones. However, the picture is not as simple as that, because our asexual strain has been previously reported to be polyploid. So this means that it is 3n equals 12, and it's an allopolyploid, which means the extra copy of genes, it's not coming from the same species of a duplication, but it's probably coming from a different organism that it's located in between the panagrelymous and propanagrelymous organisms. And what happens when there's ploidy? Well, a process known as gene conversion can occur. This means that if a deleterious mutation happens, it can be replaced by an homologous in another sequence, and this has already been reported in prokaryotes like amoeba. Now, this brings an interesting question, which is how are mutation rates, ploidy, and parthenogenesis related? I could not find a lot of information on the topic, but I found something on yeast. And here, what they found out on the study in 2015, it's that asexually reproducing yeast that was tetraploid could obtain more beneficial mutations than other yeast that were either haploid or diploid. But what does it tell us about our system? There's still a lot to learn about it. So in general, we can say from my talk that we found a lower mutation rate on the asexually reproducing strain. However, we would still like to know if ploidy is aiding maintenance of heterozygosity and preventing Müller's richet on these asexual strains, and also understand how ploidy and mutation rates are related on asexually reproducing organisms. And with that, I would like to end my talk. These are my references, and thank you for listening.
See my screen correctly? Do you see my screen correctly? I don't see.
Yes, it's working.
Yes. So I don't see your workings. So good morning, everyone, or good afternoon, sorry. Today you will have a little bit unusual talk because you will have two speakers. Nathanaëlle will focus on the genomic perspective, and I will talk about my histological findings. And together we're going to introduce you our finding on autopsodagamy in Mesophytic species. So first of all, I would like to present you the reproductive system of this species, and more specifically of Mesophytic bellari. So Mesophytic bellari can produce two types of oocytes. Male here in green are amphimectic and are produced asexually, which means that the meiosis is canonical and undergo 2 rounds of cell division. The haploid, the oocyte here is haploid, and the sperm DNA enters in the oocyte with its 2 centrioles here and here, and then the 2 haploid genome fuse in order to restore diploidy. On the other hand, female in gray here are genogenetic or pseudogamous and are asexual, which means that here the meiosis is modified, only 1 polar body is produced, leading to the production of a diploid oocyte. The sperm DNA does enter in the oocyte but remains condensed and is never used in the zygotic DNA. Only the 2 centrioles of the male are used for the first zygotic division. In addition, in this species, the prediction of oocyte is strongly biased in favor of female. Indeed, there are 90% of female against only 10% of male. Furthermore, only Y-bearing sperms enter in the oocyte. So in this species, males are produced sexually, females are produced asexually. There is a strong bias in favor of sexual sex ratio bias in favor of female, and males do not contribute in any gene to female progeny. So of course, these types of systems raise a lot of questions, but today I will only focus on how asexual females are produced. So what is happening during the meiosis of genogenetic oocytes, and what are the consequences on the genome of this species. So first of all, what is the canonical meiosis? So as Tanya very well explained, on my representation, homologs are represented in red or in blue, and sister chromatid in nuances of the same color. So after replications, homologs pair, recombination occur. During the first stage of meiosis, homologs segregate from each other. And then in the second round of cell division, sister chromatids are separated, leading to formation of a haploid oocyte. So how can we produce a diploid oocyte? In the literature, there are many hypotheses on how we can produce such type of oocyte, and 3 can be applied to the system. The first one is apomixis. So here there's no recombination, and in the mature oocyte, we find 2 non-sister chromatids with no recombination. The second option is that meiosis 1 is abortive, which can be also referenced as automixis with central fusions. This time there is some recombination, and in the mature oocyte, we find once again non-sister chromatids, but they may have recombined. The third hypothesis is that meiosis 2 is abortive this time, or automixis with terminal fusion. Once again, here there are maybe some recombination. And in the mature oocyte, we find this time sister chromatids. So as you can see, these 3 types of scenario have very different impact on the genome, and these impacts are very significant. So in order to better understand the consequences of orthopoxogamy on the genome of mesoadapted species, cytological evidences are necessary to refine genomic data. And mesoadapted species are ideal models for that, because the gonad of mesoadaptists have a stereotyped spatiotemporal organization of the gonad for the prophase 1. So at the same position, you will find every time the same stage. And concerning the other stage of meiosis, you just have to fix late oocytes, like on this image, where you can see tubulin in green and the DNA in blue. And as you can see, the 2 chromosomes here are in metaphase 1. So what is happening during the meiosis for genogenetic oocytes? In Mesoadaptive Villegas, there are 20 chromosomes. And if you take a look at this image, you will see 10 little units. This means that all chromosomes are pairs. Furthermore, these chromosomes have a cross-shaped structure, which is a manifestation of crossing over in the species. So to resume, in the species, all chromosomes are paired and all chromosomes recombine. Furthermore, if you take a look at these little crosses, you can see that they are slightly asymmetrical. This means that the crossing over are localized in the center or in the middle part of each chromosome. Now, if we go further in the meiosis, during metaphase 1, the chromosome pairs align on the metaphasic plate. Then the homologs try to segregate from each other, but not successfully. And whole chromosomes realign on the metaphasic plate during metaphase 2. Then in NFS 2, the sister chromatids segregate from each other. You can see 2 plates of 20 chromosomes here, and half of the sister chromatids are extruded in polar body, as you can see here. So in this species, the meiosis 1 is abortive, leading to central fusion with assortment of non-sister chromatids. In summary of this part, in genogenetic oocyte, DNA is only provided by female. All chromosomes recombine. Crossing over are not restricted to septonomic regions, and meiosis is altered by central fusion, leading to assortment of non-sister chromatids. So on the long term, we should expect the chromosome to be widely homozygous, depending on the localization of crossing overs.
Okay, so I'm taking over for the genomic part. Can you see my screen?
Yes.
Yes. Okay, so an important point of this genus of Mesoadaptists is that we have asexual species and closed sexual species. And so I'm going to talk about more precisely about mesorhabditis and is close sexual relative mesorhabditis spiculigera. So the first question was, is the heterozygosity of mesorhabditis bellari particularly low? So just to give you an order of magnitude, I show you here two asexual species, the Timera insect, the stick insect that Tanya spoke about before, and which have a rate of heterozygosity close to 0, and at the opposite, the ribbon worm, Linnaeus, which have an incredible rate of heterozygosity of 10%. And so our two species, Mesorhabditis bellari and spiculigera, are in the middle of the graph with 1% for Mesorhabditis bellari, which is lower than spiculigera, but is not very low. And if we compare to other species with other mode of reproduction, these two species are in normal range of heterozygosity. So here we have high rate of heterozygosity, but recombination in an asexual species. So one hypothesis could be that we saw chiasmatas, but there is no recombination. So to test that, we estimated the recombination using the linkage disequilibrium. So when there is recombination, there is no linkage disequilibrium, which means that if you have two positions along a chromosome, if you know the sequence at the position 1, you cannot guess the sequence at the position 2, so you can have all the combination in individuals. However, if you have no recombination, these two positions are transmitted together to the offspring, and so you can guess the sequence at the second position if you know the first one, and you can find only one or two combinations in the individuals. To estimate the recombination by this way, we need several individuals, the two haplotypes for each individuals, and a computational way to estimate the recombination rate from these data. So to achieve that, we sampled 10 individuals from 10 locations and we sequenced genome. We then did haplotype phasing because you have homologous chromosome, but by doing simple genome assembly, you obtain a single consensus sequence. And the haplotype phasing allows us to recover the two haplotypes in two different assemblies, which allow us to compute the linkage disequilibrium and estimate the populational recombination rate using a Bayesian software. And so this recombination rate depends on the effective population size and the recombination rate. So what we obtain for the sexual species where we expect to have recombination. So here in y-axis you have the recombination rate, and in x-axis the position along the chromosome. So for this species, you can see that first there is no hotspot of recombination, and the mean value is around 0.4, which is really high because for most species, the value is 10 to 100 times lower than this. And this suggests a very large effective population size. And so now for our asexual species, Mesorhabditis bellari, we have a mean value of 0.2, which is half lower than the close sexual species, but it's still very high. So there is recombination in this species, and it suggests also a very large effective population size. So here we have a paradox because we have asexual species with 1,000 individuals in lab and never find any sexually produced female, and we have cytological and genomic evidence for recombination. However, we have a high rate of heterozygosity. Something that can help us is to look at the distribution of the heterozygosity along the chromosomes. Why? Because if you have recombination with the maternal chromosome, you can have 4 different types of gametes. 2 types of gametes which keep the heterozygosity by keeping together the 2 ancestral chromosomes or the 2 recombining chromosomes. or you have two gametes that lead to a loss of heterozygosity because you have one ancestral chromosome with one recombining chromosome. And so if we look at the heterozygosity along the chromosomes, in this case, we should have a stable heterozygosity all along the chromosomes, while here we should have a normal heterozygosity for the first part, but for the end of the chromosome, we should have an heterozygosity of 0. So we measure the heterozygosity along chromosomes for the 10 individuals we sequenced. And what we obtain is for one individuals, we add 20% of the contigs where the part of the contig has a rate of heterozygosity of 0, and the second part has normal rates of heterozygosity. However, for the 9 over individuals, the heterozygosity is stable along the chromosomes for all chromosomes. So, in lab, we observe only asexual reproduction, but this result suggests that maybe there is indeed asexual reproduction, but also sexual reproduction. But sexual reproduction seems, or rare sexual reproduction seems not enough to explain this pattern, and so maybe there is other mechanisms that allow to maintain the heterozygosity. For example, we could only keep these two types of gamete and remove these two one to keep the heterozygosity high. And so finally, to conclude, we can say that the cytology allows us to understand how the diploid is conserved in genetic embryos, and it validates the presence of recombination and allows us to estimate where the crossovers occur along the genome. We finally detect in vitro and genomic evidence for recombination, and this recombination affects all chromosomes. But finally, the genomics reveal that they probably still do sex, maybe very few, which explains that it's not observable in the lab. One other hypothesis is that sex occur only in nature and not in lab because maybe stressful condition. Or maybe the rate of sex could be different between the population because we have 10 individuals from 10 different populations. And finally, we could have mechanisms allowing the maintenance of heterozygosity. And to finish, I would like to thank you all the people who participate to this project, and especially Cécile Di Blasi, who did all the analysis of recombination rate. And thank you for your attention.
Hello, everyone. I'm Shan, a PhD student from Bast lab at the University of Cologne. Today, I will show you our study about a new high quality and phased single individual assembly from ancient asexual orbit mites, Platynothrus peltifer. Firstly, I want to provide questions of sex. So why sex is maintained in nature? Most of MUTATIONs produced sexually, but sex is very complicated and costs a lot, such as producing males. But in asexual, like in personal genesis, they don't need males to produce them. So no sex, no cost. But sex doesn't have any advantage here. Nowadays, people think predicted that sex has 3 big advantages in evolution. That can mention the genetic diversity and validate the effect selection. Moreover, sex is also seen to be need for long-term persistence of species. And also the asexual is regarded as a dead end in evolution. Despite sex has been considered as a requirement for species persistence, but there are still some ancient asexual schedule. Organism possessed hair and devices for million of years, like deluded, rotiful, and orbited mites. They are called ancient asexual schedule. The dialect rotiful you are used for the research about asexuality in last decades. But a few years ago, research found some evidence for recombination. So there must be some kind of cryptic sex between them. So we chose the orbited mites to be a model for our study. Orbited mites are very easily to get and in forest, and most of them are asexual. And the biggest advantage of orbited mites as model organism is that they provide evolutionary replicates. They have 159 genome and about 10% of species are asexual. So they make it possible to compare the sex and to asex. Prevalence study on various asexuals allow predictions of potential consequences of ancient asexuality. People find that allelic divergence over time of sex and recombination, but they use few phased regions, so it's hard to say how extensive. And the asexual mentions effort selection in themselves, but we don't know the population size for them. And Dr. Jans in our lab also found that transposable elements inactive and low abundance. But he did that with unfazed and low quality pooled sequenced genome. So the lack of the high quality phased genome from single individual becomes a bottleneck for our study. For the future study, we take a closer look on Platynothrus peltifer. It's an ancient asexual schedule which divorces and have distant macrolines, which resemble the outcome of the geography draft. And they also very old, up to 300 million years. And they are very easier to available and to compare with their close sexual relative like Hamania. I will talk about that later. This is our sampling place in Eifel National Park Forest in Germany. And this is the place, simply spot of us, and this is me. Before we assembling, we established a DNA extraction protocol for ultra-low important organism because the mites are so tiny. This built by Hüsna in our lab, and thank you for Hüsna for this. That make us our single individual genome assembly become possible. And then we combine the Pacabell-Hi-Fi and the Tell-seq method for the reference sampling, and Tell-seq also for the others for additional population data. They're also in single individual. Now this is our genome assembling pipeline here. In the red square, that's what we done now. We use PEG-BEL for the de novo assembly and then use a Tell-seq long linked rates to improve the assembly. And now we are in the improved assembly this step. Vectors 2 also used for the contamination remove and the Busco and the Kit software used for the assembling quality. Genome scope analyze realized that Platynothrus with diploid genome structure and with 2.4% heterogeneity here, and the genome size of Platynothrus is about 170 megabase. If we took a look at the smart plot on the right, you will see the genome is infected del plot. Finally, we got the high quality and fixed assembly with N55. 50 is about 8 megabase with 15.5 scaffolds here. This is our primary assembly, and we also provide the alternatives assembly here. This contains some allelic loci and some unplaced scaffold here. The cat analyzed on the left shows the separate haplotypes. The black space are the alternative alleles, which are in our alternative assembly I showed before. But here we just present the primary assembly here. The Busco analyze use two databases that eukaryotic and arachidid database for use a single copy gene to detect the completeness of our Platynothrus genome. They are both in 97%, about 97%. So our genome is completeness. Vectors results show our assembly with a low contamination. So our protocol we build to extract our DNA is good for use. So you can see here just a very few scaffold in the yellow square show quite high GC content. But we didn't remove that before because we want to left them for the further analyze like a horizontal gene transfer, or I will talk that later. So after the genome assembly, we also have some analyze on the population like in the later. And we also have some questions here, try to answer. Is about the structure virus, including the horizontal gene transfer in Platynothrus. And also we want to detect that there is some cryptic sexual between Platynothrus. And we also want to do the demography analysis for Platynothrus and the transposable element active. Before the horizontal gene transfer, we use the previously published transcriptome to annotate our assembly. We annotated about 4,000 genes, but we accepted for 15,000 genes in mice. So we also sequenced our own RNA-seq data, but we are still waiting for them so we can get a better version of annotation in the future. But now, nevertheless, HCT analysis results show here. We found about 1.5% genes show HCT. Normally in asexual species is about 2%. So it's okay here. And we also provide a sample here that tamanasm is a kind of enzymes. People found that before in fruit, a kind of plant is thought to offer protection from insect. And this gene stems from the bacteria. We also show the landscape of the transposable element here. It's contrary to previously low abundance and inactivity TE detected. Here we can see that about 23% TE activity and active and 3 post burst. And this is maybe a recently activity TE post burst. This analysis conducted with the EDTA software and repeat masker. In the 5 individuals we got from the same sampling spot, they divided into 3 lines, and we also calculated the nucleotide diversity on all sides. Here the pi is about 1.344%. It's not high and not low, so it's okay here. And the LD analysis of Platynothrus, we found that there are no recombination here in Platynothrus. So our Platynothrus is indeed obligate asexual species. And the 4 gamete test also provided a similar result for us. So our method is asexual. The demographic analysis of Platynothrus show they have a large and stable population size compared to their sex relative called harmonium. Here they are sexual. You can see our asexual Platynothrus have a larger population. Okay, so take home and outlook. Orange-based methods are good model to study asexual genomics features after our study here, and we provide a high-quality fixed Platynothrus peltifer genome for future study, and we also established a DNA extraction protocol for ultra-low input. Maybe you can use that in our other tiny organism. And we also found that no correlative sex between the Platynothrus, so we are the totally asexual organism, and they have a large and stable population to help them to accept the extinction. And we also find some different results in TE active, different to be previously studied. That's our result here. And next, we will continue compare different asexual to sexual relatives to find out the true consequences of asexuality. Thank you to Bast Lab at the University of Cologne. Thank you, Dr. Jens Bast, Hüsna, Svenja, Clara, Nico, Christine, and Linda. That's all. Thank you for listening.
So hello everyone, I'm Cécile Molinier, and today I'm going to present you some work I did during my PhD with Christophe Hagen to my Blanc. So the genomic consequences of asexuality are often studied by comparing asexuals found in nature which are generally clonal, with closely related sexuals, as you can see in this putative phylogeny. However, these comparisons may be biased by selection because asexuals sampled in nature are only a selected subset of the most successful lineages, and so we lack the failed ones. So they contain limited, or at least not all, the information of how asexuality evolved in the first place. Moreover, because asexuals have evolved from sexual relative, they generally uncover different types of meiosis modifications, more or less advanced toward a system that can be genetically equivalent to clonality. But considering this cellular process in such intermediate steps is important because selection would differ with respect to recombination and segregation on them compared on pure clonality. So for instance, here I'm going to show you the two types of asexuality mode of reproduction derived from meiosis with two opposite genetical consequences. So some mechanisms can lead to a complete loss of heterozygosity, which is kind of the terminal fusion type. So I would abbreviate in my talk LOH for loss of heterozygosity. And but because recombination can still take place in the first stage because it's derived from meiosis, we could find some heterozygosity retained as far as we go from the centromere. Other mechanisms can lead to the opposite genetic consequence, which is a complete loss of heterozygosity. And this is the one we found generally in nature, with the offspring cells being equal to genetically to the mother cell, so with a complete loss of heterozygosity in absence of recombination. But again, if it occurs in the first stages, we would thus have some homozygosity in regions distal to the centromere. So here is just to show that it's possible to have a huge diversity of asexual mode of reproduction, and maybe these are found in intermediate stages, but because they are rare in nature, we generated new asexual lineages. So to do so, we used Daphnia-Culex, which is a microcrustacean that has two distinct reproductive modes, a cyclical parthenogenes, in which female clonally produce daughter or son via an aborted meiosis, so it's derived from meiosis, and a sexual reproduction which leads to the formation of a diaposal egg called ephipia. And today I would be interested in how these ephipia are produced. And in this case, because it's produced sexually, I would call these lineages the sexual one, whereas other lineages are obligate parthenogens. This means that they always reproduce asexually, and this time females still produce ephipia, but they do not need to be fertilized by a male to give rise to new female of the same genotype of the parent. Moreover, we know that these asexual lineages are still able to produce males, we call rare males, that thus can mate with sexual females and transmit asexuality into new lineages, which is this process is called contagious asexuality, and it's what we used to produce our new asexual lineages. So how we did that? Thanks to the existence of another completely independent phenotype, which is called the non-male producer, which would be in circles in my presentation. We were able to use a mass mating approach. So we selected a sexual non-male producer lineage that we let it reproduce by its asexual part in a big aquarium. And if this female produces ephipia, this one would be empty because there are no male to fertilize them. So this is important. In the non-male producer phenotype, a female can only produce full ephipia only if she's asexual. But because here it's sexual, this ephipia would be empty. And then it's when we manually add rare males, where asexual males, into the culture that allow us to be sure that any full ephipia would come from an event of sex-asex cross-mating. So then, to investigate the inheritance of asexuality, we studied the reproductive mode of the F1 hybrid produced by looking at the segregation of 1 to 4 microsatellite markers in their progeny, which are these little embryos you can see here contained in the ephipia they produce. So now I'm going to show you the result of one single sex-asex cross. So here we have the F1 hybrids, 5 non-male producer and 17 male producer F1. And we can see that in the y-axis, we have the loss of heterozygosity rate of the offspring in the offspring of the F1. So again, I put the asexual natural lineages we have, which contains the parental asexual lineage we use for this cross, which are in yellow. And indeed, we can see that this asexual from natural population reproduce clonally with a loss of heterozygosity rate equal to 0. The second reproductive mode we should expect is a sexual reproduction, which leads to a 0.5 loss of heterozygosity rate, because within a lineage, sexual reproduction is equivalent to self-fertilization. And so you can see here that for the F1 hybrids, the huge diversity of the loss of heterozygosity rate cannot be explained only by clonality or by sexual reproduction. Moreover, you certainly noticed that the only phenotyped F1 hybrids are the non-male producer. This is because, as I just said before, the non-male producer cannot produce ephipia that contain embryos unless they are asexual. So we are sure that these clones, the embryos we collected in the ephipia of these clones are produced asexually. And when we looked at the loss of heterozygosity rate they have, this could be explained by the two types of asexuality modes that I showed in the introduction, which is a complete loss of heterozygosity without recombination for him, a complete retention of heterozygosity for this clone, and also maybe one or the second mode of reproduction with recombination for the intermediate loss of heterozygosity rate. So all the asexual mode possible. On top of that, we know that at least a part of this F1 non-male producer in circles are capable of doing both. They are capable of producing offspring asexually, but when we add males to them, they can mate with them. So this was checked with paternity tests, and so we also this also lead to some loss of heterozygosity rate average over all the offspring, something more toward 0.5, because we have now a mixed reproduction, which was never found in Daphnia before. However, this also renders the phenotyping of the male producer more difficult, because the male producer can produce male, and so we would not know if the loss of heterozygosity rate we have on their offspring is coming from an event of asexual or sexual or mixed reproduction. So we analyze this data. So it's the same data I'm showing there with the F1 hybrid and the loss of heterozygosity rate we found in their progeny using models. And so we were interested in estimating P, which is the proportion for a given F1 hybrid of producing offspring sexually, which lead to 0.5 loss of heterozygosity rate. And again, the loss of heterozygosity rate we wanted to estimate of the asexually produced offspring. So by comparing several models, we were able to show that the offspring of the male producer F1 hybrids have some loss of heterozygosity rate that couldn't be explained only by sexual reproduction. And so we have also some mixed reproduction going on this male producer. And there was no male producer or non-male producer effect. The best model to explain our data is the one where all F1 hybrids do 69% of their progeny sexually, and the rest of them asexually. And the loss of heterozygosity rate of this asexually produced offspring among F1 hybrids is very, very variable, with, again, most of them that are not clonally produced. As you can see here, the 0 would be a clonal mode, asexual mode of reproduction. And again, with some loss of heterozygosity rate indicating some recombination taking place. However, these models are still an ongoing project because in our data, we lack one category, which is this sexual non-male producer, which we are now integrating to estimate S, the proportion of the F1 hybrid that produce offspring only sexually. So, this is something which is ongoing, but this will not change the main result of mixed reproduction and also the huge variability of loss of heterozygosity rate reflecting different asexual mode of reproduction. The second thing that I would like to talk about is about the fitness of this newly generated asexual. So first, we in fact had several issues because the hatching rate success of sexual-asexual crosses were very, very low compared to the sexual interpopulation crosses, which were our control. So for instance, here, based on a little example of 25 embryos, we see that the sexual-asexual cross had very, very low hatching rate compared to the sex-sex cross. But we are now acquiring more data to elevate the sample set and also on different sex-a-sex and sexual-sexual crosses. And second, the contagious asexuality led to strongly reduced fertility rate in hybrids compared to natural population. So on this figure, you have here the fertilization rate, which is the amount of embryos we find when we open the epipia. And so the gray plus the yellow points show the distribution of this fertilization rate in all F1 hybrids across all sex-a-sex cross and in all natural lineage we have. So this distribution were not significantly different, but when we looked only at the asexual ones, so in the hybrids compared to the asexual natural lineage, we see that this difference was significant. That means that the asexual ability of the F1 hybrids seems to be much reduced compared to the asexual ability of the asexual found in nature. So altogether, these results suggest that the new asexual lineages we produced here by a contagious event differ from those found in nature, from the asexual found in nature, both in terms of the asexual fitness and also in terms of the asexual phenotype. So we would not have these two discrete classes of asexual and sexual, but more something more diverse, which is non-binary, with very different asexual mode of reproduction, recombination, and mixed reproduction. And so maybe this would this new asexual lineages formed would then be subjected to subsequent selection, for instance, based on their fitness or the genetic loads they can carry, depending on the asexual mode of reproduction and also depending on the recombination rates going on. And so then we would find at the end these two discrete classes and selection for one special type of asexual, which is equivalent to clonality. So I would like to thank you and to thank my supervisor and the students that helped me for the fitness part. Thank you.
Okay, good afternoon from Catalonia. Now we go to talk about planarians with our presentation, differential expression analysis in sexual and asexual freshwater planarians populations. Planarians are free-living platyrmins that can live in terrestrial, marine, or freshwater ecosystem, and the freshwater planarian are broadly distributed around the globe, being the Duhesia genus the most diversity and distributed genus in Europe, Africa, and Asia. In the Mediterranean region, this genus is broad distributed and have been widely studied. In this region, the genus shows two reproductive strategies. The sexual reproduction occurs when hermaphrodites in individuals interchange sperm between them, and they are internally fertilized with the sperm of the other partner. On the other hand, the asexual reproduction occurs by fission, and on this moment from now, we go to use a Fissiparus for a call to the asexual animals for a better understanding during the presentation. Well, and the fission occurs when an animal's device spontaneous and each fragment generates the missing structures. This characteristic is what makes famous to the planarians, but most interesting is how the group used this capability as a reproductive strategy and how they combine fissiparity and sexuality. The combination of both strategies vary between species. Some are exclusively fissiparous or exclusively sexuals, and others can show fissiparous population or sexual population, and even we can find sexual individual and fissiparous individuals in the same locality. With all this background, we have in the Mediterranean region all the possible combination of the reproductive strategies with sexual species, fissiparous species, population with fissiparous and sexual individuals. And with all this information, we want to identify the difference in the expression profiles of orthologous genes between sexual and fissiparous planarians. For that, we studied the Iberian Peninsula and Italia, and we established 4 comparison levels. We compared in fissiparous individuals with sexual individuals of Duhesia subtentaculata in the same locality, Cangas. We compared Fissiparus population of Duhesia etrusca from Latrilla and asexual population of Duhesia etrusca from Prive. We use also different species in the comparison with Duhesia subtentaculata, that is Fissiparus from Bosque, and Duhesia villaferrei, that is sexual from the same locality. And finally, we compared Duhesia subtentaculata, Fissiparus from Santa Fe, and Duhesia villaferrei, sexual from Bosque. The samples were collected from localities previously identified as sexual Fissiparus or mixed, just as in the case of Kangas. Posterior to the collected, the animals were submitted to visualization for the identification of the gonopore in the sexual animals. They were then conserved in RNA later and for posterior extraction with trisor protocol. We sequenced 20 samples and with 3 or 2 replicates by population and reproductive strategies. For the bioinformatic analysis, we use the parent reads, and these reads were filtering by quality and trimmed, and using for the assembly with Trinity. We obtained an assembly by each sample used in the study. And the transcripts were clustered with CD hit and filtered with a strategy based on the result of Brute Tools analysis. The filtered transcripts were traduced to proteinac with transdecoder, and the longer isoform were selected. These isoforms were used as input in the shares, the orthologous shares with OrthoFinder. In this case, we based this strategy in the analysis described by Parker and collaborators. From the OrthoFinder analysis, we obtained a set of single copy genes, and we captured the nucleotide sequence from corresponded with these protein sequences from transdecoded output. The single copy were aligned, trimming, and filtered by length, and the obtained set was annotated with the trinotate pipeline. At the end of the process, we obtained an annotated reference by samples of around 3,000 orthogroups. The read of every sample was mapped against the corresponding reference with Callisto, and the abundance files were imported in DSEC2 for the difference expression analysis, using the sexual condition as denominator of the fold change calculation. Because we use samples from different localities, we worked with the shared upregulated genes and downregulated genes from all comparisons. Both set of shared genes were analyzed with GeoSeq, and the result of the regiment analysis were visualized with Revigo. Finally, to look for orthogroups shared only by samples with the same reproductive strategy, we selected only the orthogroup shared by Fissiparus and the orthogroup shared only by sexuals. Both set of genes were aligned, trimmed, and filtered, and at the end, we obtained a set of annotated gene shared only by the reproductive strategy. Passing to the results, we established the differential expression analysis in all the comparisons. It's interesting that we found a less number of differentially expressed genes in the comparisons with samples from the same locality. And that results will reflect the adaptation of these animals to the different environmental conditions. We obtained 10 shaded enriched genes for Fissiparus samples and 38 enriched genes in sexuals. And if we look to the value of log for change of this set of genes, we can see that these values are heterogeneous, and the genes enriched in sexual animals show a major expression values. The gene ontology analysis shows an enrichment of nuclear component in Fissiparus with an enrichment of biological process related to cellular replication, response to stress, regulation of biosynthetic process, and muscle tissue development and cell differentiation. These results all reflect the response to stress and in Fissiparus related to regeneration process. The enrichment analysis in sexuals, on the other hand, shows an enrichment of different cellular components and biological process related to metabolic process, metabolic regulations, and response to stimulus. The enrichment of this metabolic activity pathway and response to stimulus could be related to the maintenance of homeostasis in sexuals. Finally, we didn't find any orthogroup shared only by Fissiparus samples. On the contrary, we found a lot of orthogroups shared only by sexual, and after all the process, we obtained a set of annotated genes of 119 genes shared only among sexual samples. The visualization of this annotation in Revigo showed the presence of cellular component of the sperm related to the process, the spermatogenesis process in these sexual planarians. With all these previous results, we now are working on the top differential expressed gene using a log for change threshold equal to, and the top enriched gene ontology of this term. In addition, we are working in the gene ontology enrichment of only sexual genes and in the identification of genes previously described as the core of the fission process or sexual reproduction. Nevertheless, with our previous data, we can conclude that the metabolic activity of Fissiparus planarians is focused on the cellular division and the response to the stress of the regenerative process, while the expression profiles in sexual planarians is aimed to respond to the environmental stimulus and the maintenance of homeostasis. In addition, and because sexual planarians maintain regenerative capacity in response to injuries, they could maintain the expression of genes important for fissiparity, while sex genes are only expressed in sexual planarians and are under strict regulation in Fissiparus planarians. We hope to discuss with you our results and open the debate about this topic in these interesting animals that are planarians. Finally, I want to thanks to our research group from University of Barcelona, our collaborators, and you for listening to us today, and to the planarians, of course. Thank you very much.
All right. Can you see my screen?
Yes, perfect.
Okay, great. Yeah. Okay, so first of all, thank you very much for Astrid and Philipp to organizing this exciting symposium. I'm Wen-Juan. Currently I'm postdoc at University of Kansas. I'm mainly interested in evolution of sex chromosome, sex determination, and asexuality. So today I will discuss some results. Actually, it's my final PhD chapter for the PhD thesis, which has recently finally published. Took a couple of years. So it's a big relief for that. So I'm talking about mainly the genetic and also genetic basis and also evolutionary consequence of this female sexual trait decay in an asexual wasp. So I just need to change a bit here. Right, so trait decay may occur when selective pressure changes. So due to either environmental or lifestyle changes. So this would lead to, say, former maybe adaptive traits become non-functional or even more adaptive. So the trait decay is very widespread across taxa, so they can be found in various, say, morphology, also in physiology and behavior, etc., etc. And so similarly, we also can expect that so sexual traits may decay following the transition from sexual to asexual reproduction, as also Tanya has gave brilliant introduction there. The selective pressure also changes. So males are no longer produced and also males are not exposed to selection under this asexual reproduction. So indeed, for the male sexual traits, which as they are no longer exposed to selection, are not produced, so if they decay, they may decay as consequence of drift. So in fact, this review conducted a couple of years ago, so they have combined a variety of taxa. They actually showing that most male sexual traits are largely remaining or fully even functional in asexual species. However, in sharp contrast for the female sexual traits, such as the mating attraction, courtship behavior, egg fertilization. So these types of traits actually undergo through a very rapid decay. So maybe it's not surprising given these traits are quite costly to maintain them. So actually also has been in this review, they also found extensive decay for the female sexual traits. So here I'm just focusing on one trait, in particular this female functional virginity mutation. So this occurs when this mutation, which causes female have reduced mate attraction or also reduced copulation probability or their behavior, or even reduced if they actually mating, reduced egg fertilization. So this trait is quite interesting to study in this Hymenoptera insects. Cause this Hymenoptera insects have this haploid-diploid sex determination system. So meaning that females are all developed from fertilized eggs are diploid, and the males are haploid and developed from unfertilized eggs. So if this mutation occurs in these haploid-diploid systems, so you can imagine that, so this would cause the females produce exclusively these haploid strands. So we can easily to measure and also to various crosses, you can track them. So another thing is found that actually this mutation is favors during this early spread of the facultative asexuality. So during this early stage, so they have highly female by sex ratio. So this would select for females. In this case, they would produce more signs when these mutations occurs in these females. So also here is showing a model a couple of years ago by Salsamer. So actually just ignore the details. So basically they actually modeling this asexual is induced by some endosymbionts. So hence here is infected. So here is focused, so what they found actually after, say, 20, 40 generations. So these homozygous, this is female functional virginity mutants, they actually can spread and also increase the frequency and getting fixed in this population. However, this underlying genetic or molecular basis of this trait or mutation is remain largely unknown. So it's also probably historically, so it's difficult to conduct genetic courses or any mapping studies in asexual species due to this lack of recombination. However, we could solve this by working on this indocin-bound-induced asexual Hymenopterus. So in this case, we could using the antibiotics to cure this bacteria, and then we can partially restore this sexual reproduction. So in this case, they would produce asexual females would produce asexual males, and then we can because asexual males are largely actually functional, so you can cross to the sexual female backgrounds, and you can cross probably for more generations. So here I work on using this system, which is a quite ideal system to address this question, is Asabara japonica. So this is a parasitoid wasp, which parasitizes the Drosophila melanogaster in the second instar larvae, and then they work on biting. So if that's successfully, they will develop a wasp. So this species also have segregation between their asexual populations and sexuals. So asexual mainly distribute in the major islands in Japan, and in the southern, the small islands, they have several sexual populations there. Furthermore, the species, the asexual parthenogenesis is induced by this indocin-bound called bacteria, bacteria. So for this project, I'm particularly interested question to address is to understand first what's the genetic architecture of this female functional virginity trait, and furthermore, we want to understand if we can identify these regions, what kind of genes are underlying for this trait. So first of all, of course, we assess these phenotypic traits for this sexual trait decay. So we have asexual and sexual females within the same species. So we assess several traits like female sexual attractiveness, the mating behavior, their also the mating success or not, and also their spermatozoa, which is the organ which in the females, so they can store the sperm whenever they decided to fertilize them. So we found that compared with the asexual females, compared with the sexual females, the asexual females, they have reduced sexual attractiveness to males. They also have almost complete the mating success. Then when we give them the single pair mating opportunity, we don't find any sperms found in the spermatozoa in this asexual female. So we conclude that this female functional virginity is likely due to this complete mating failure in asexuals. So then we want to monitor these traits throughout various generations. So in this case, we conduct a series of crosses. So we cross asexual genome to the sexual females, and then we back cross for a series of 4 more generations. So within the crosses, you can see in this dark here showing that increased asexual genomes introgressed in these introgressed females. Then we first see this phenotype of this trait. Just to remind you that, so in these haploidiploid systems, so if this mutation or this trait evolved occurs, so we can, they will produce haploid strands only. So in this case, on the y-axis, I actually monitor the proportion of females actually showing this functional virginity trait. So as you can see that when we increase the asexual genome in F1, we actually showing no difference with compared with the control, which is the sexual females. Then furthermore, when you introgress more these asexual genomes in the 2nd and 4th generation, we found a significant difference increase for this female functional virginity we scored, and it's roughly 50% in both generations. So we also conduct a very simple model which involves the 3 unlinked loci, try to see which can better explain our data. So our model actually showing that our data is consistent with a single locus model with recessive effects. So at this point, we get all the phenotypes. So we want to basically figure out what the genetic regions involved. So we want to do the classic QTL analysis. So this is non-model organism. So there's no genetic marker available by then. So first we need to conduct a genetic linkage map. So we did F2 crosses. Then we're using this mainly the recombinant haploid F2 males to conduct a genetic linkage map. So I will skip the details for the pipeline, but briefly, so we have a low coverage of short-read Illumina sequencing for both sexual and asexual populations. And then we are using a pipeline which don't require your reference genome. Then we go through a series of stringent, very stringent the filtering steps. We get a series of SNPs. We also validate them in the wet lab. So here is the number basically we finally use for our linkage map. Then we conduct a genetic linkage map. So this is the final, the linkage map. So this is composed of 16 linkage groups in total and span over 600 centimorgan. On average, it's about 7 centimorgan distance between SNP markers. So our linkage group is consistent with the karyotype which found in this species, and also the linkage map is very compatible with other published Hymenopteran linkage maps. So furthermore, we then using this genetic linkage map to conduct this QTL analysis. So here, as you can see that the x-axis is 16 linkage groups, and the y-axis is ROD. So ROD is to make likelihood to measure the possible association between any the genetic markers associated with the trait which you are interested. And so anything above this 5% threshold, they showing significant association between these markers and also the traits. So above the 5% the ROD threshold, so we detect a single big QTL in the linkage group 2. Regardless of any algorithm we use, they always detected in a similar regions in this linkage group 2. So this single QTL can explain up to around 49% the phenotypic variation. So furthermore, among above this 5% threshold, we didn't detect any other significant QTL throughout the whole genome. And we also didn't find any interactions for QTLs, detected QTLs. So after getting this QTL, we want to understand, so what's these regions under this QTL? So, sorry, one thing I forgot to mention. So this QTL, within this 95 confidence interval, we found these 2 SNP markers associated with this QTL, and we further look into this possible the association with their mating success. So we found in both markers regions, so if they are heterozygosity, so they have highest mating success. So if they are homozygous, so they are very low mating success. So furthermore, so we want to understand what these regions underlying the QTL and the possible genes involved. So we then did a candidate gene analysis. So in this case, we did a PacBio genome assembly, de novo genome assembly. So here is just some stats for the genome assembly. In fact, we Bootstrap score showing rather complete the genomes, which has been quite good in, well, not compared with others, but at least within this Hymenoptera insect species. So we then, using this PacBio genome, we took these QTL marker regions, which they flank in the DNA fragment, then we can map blast to the PacBio genome. Then we found actually this, we can detect 2 unoverlapped contigs, which is covering more than at least 4.2 meg-based regions. So furthermore, we also using, we have ovary PacBio RNA-seq. We also have using some other published Hymenoptera transcriptome. So we further annotated these QTL regions. So we found at least the QTL regions contains at least 131 coding genes. So then we hypothesized that, so because this trait mutation, female functional mutation trait is related to female attractiveness and mating reluctance. We think that this might underlying differential gene expression in the sexual species. So then we conduct a differential gene expression, sex-biased gene expression in their sexual sister species as a biotarget. So basically because we're using a whole body tissue, we're using very stringent criteria to call sex bias. And also, of course, FDR corrected. Then we further, among this, under the QTL have 131 genes. Then we found that 15 of these genes show differential gene expression, and most of them showing female biased gene expression. So here are the list of all these 15 genes under the QTL, also showing sex bias. So 3 of the genes I'm highlight a bit. So compare with other related, compare with sex asex studies. So we can find, so this is CD151 antigen. This gene is transmembrane family 4. So this is involved in cell signaling. So this gene has been found in this differential gene expression come up with the list between the sex and asex Tamimastic insects by Darren Parker and also in Tanya's lab. So another gene is this NUP50. So this gene is involved in the mRNA transport. So this gene actually has found accumulated deleterious mutations in this asexual genome when they compare the sexual asexual genome to see what genes accumulate deleterious mutations in this Leptoplana clavipes parasitoid wasp. And finally, there's another gene, hormone receptor 4. So this gene is gene code for hormone receptor 4. They found that in some Drosophila species, this gene actually have medicated precise timing for the hormone, steroid hormone. And also a recent study actually found in the malaria mosquito species. So they found that after the male actually can transfer the steroid hormone to females. So after the transfer to females, maybe they interact with the steroid hormone. Actually, this leads to a complete loss of female the receptivity to mate with other males. So it's quite interesting related to a mating reluctance and rejection. So we also found in our studies, I don't give the details. So we also found the premature subcodons for these genes. We also found increased SNPs in the promoter regions for this gene. So just take home message for this talk. So we found perhaps this complete mating failure leading to the female functional virginity in the asexual in the species. So we also conduct genetic linkage map for asexual species and various genomic database, which be hopefully be useful for other comparative genomic studies across Hymenoptera species. So we found these trees actually have a very huge single QTL with the large phenotypic effects. And under the QTL, we found up to 131 candidate genes. 15 of them show sex bias gene expression, and 3 of them are perhaps top interesting genes, which will follow up molecular functional studies. So with that, I'd like to thank all these co-authors for all the help throughout these quite some years to finally get it published. And my PhD fellowship and the fundings and also competition from various clusters. And also the former lab members, collaborators, and others, and all of you for your attention. With that, I'd like to take questions. Thank you very much.